The Science and Art of Clinical Genetic Variant Classification and Its Impact on Test Accuracy, published in the Annual Review of Genomics and Human Genetics. The authors are Hunter H. Giles, Madhuri R. Hegde, Elaine Lyon, Christine M. Stanley, Iain D. Kerr, Megan E. Garlapow, and Julie M. Eggington. Authors Giles, Garlapow, and Eggington are employees of the Center for Genomic Interpretation based in Sandy, Utah. Author Madhuri Hegde is an employee of Perkin-Elmer Genomics based in Waltham, Massachusetts, and holds an appointment in the Department of Biological Sciences at the Georgia Institute of Technology in Atlanta, Georgia. Author Elaine Lyon works for Hudson Alpha Clinical Services Lab in Huntsville, Alabama, and author Christine Stanley works for C2I Genomics in Cambridge, Massachusetts, and Variantics in Framingham, Massachusetts. Author Iain D. Kerr is a consultant from Ann Arbor, Michigan. Abstract. Clinical genetic variant classification science is a growing subspecialty of clinical genetics and genomics. The field's continued improvement is essential for the success of precision medicine in both germline, hereditary, and somatic oncology contexts. This review focuses on variant classification for DNA next-generation sequencing tests. We first summarize current limitations in variant discovery and definition, and then describe the current 5- and 4-tier classification systems outlined in dominant standards and guideline publications for germline and somatic tests, respectively. We then discuss measures of variant classification discordance and the field's bias for positive results, as well as considerations for panel size and population screening in the context of estimates of positive predictive value that incorporate estimated variant classification imperfections. Finally, we share opinions on the current state of variant classification from some of the authors of the most widely used standards and guideline publications, and from other domain experts. 1. Introduction. Clinical genetic variant classification science is hard. Compared with the vast array of other clinical science specialties, genetic variant classification science is in its infancy. The expanding community of variant classification scientists is progressing through inevitable growing pains, while learning from both its successes and its unfortunate failures. Although the science of clinical genetic variant classification still has years to go before developing into a mature clinical science, some major breakthroughs and lessons learned from mentors in older clinical science specialties have yielded initial progress. Variant classification scientists have become a significant strength for supporting diagnoses and have already contributed to the implementation of precision medicine, particularly preventative interventions and targeted therapies. As we learn more and remain focused on raising the bar, clinical genetic variant classification scientists expect to continue contributing to the advancement of diagnostic, predictive, prognostic, and theragnostic medicine. This non-exhaustive review discusses the legacy of genetic variant classification science and how it affects today's methodologies, primarily in the United States. Commentary from domain experts will help us to look both backward and forward, with a focus on opportunities to improve the accuracy of the field in the coming years. Note that at the time of this writing, major efforts are underway to revise the societal variant classification guidelines for both germline and somatic methodology. Therefore, some of the issues raised here may be in the process of being resolved when this article is published.